Okay. So after going through the exams, all questions and all parts as well, taking into account how long everyone spent on each section, and also the averages for each section. This is the score distribution that we have for the course. So the average is 51, which is a little bit lower than normally for this course, at least this particular section. Other sections are, this is probably the average for some other sections, depending on who the instructor is. And so, you know, and as usual, more than half the class is usually above average. This is kind of very similar to what I've seen in previous years as well. So if I compare this to previous years, and this is kind of the average for previous years, depending on whether I teach it in the winter or the spring quarter, it doesn't really matter. So this is kind of the average that we see. So we're running 10 points below the average. And that could be contributed perhaps to, you know, as many of you mentioned, you didn't have enough time to finish part C or carefully. So it could be a result of that, or it could also be a result of just doing this type of exam online, or the way that the questions were structured. But in general, the exam is definitely very similar to previous years in terms of the number of questions, which usually can be done in the amount of time. But we do recognize that being online, it could be a little bit, it could slow everyone down. I mean, it slows down many people even just teaching the course online, right? Just the pace is a little bit different, so we can take all of this into account. So to account for the lower than usual average, the option that we've decided after multiple discussions with multiple other professors who've taught the course in this online format is to allow you guys to increase this exam 1 grade by giving you an extra credit assignment. The assignment will be worth 10 points. It will cover pretty much the material from lecture 1 to lecture 6. It's going to be very similar to part C, right? So since that's the one that most people weren't able to finish, I will deploy it on Monday. You have an hour to do it, to complete the assignment. So you'll see the timer telling you an hour, which is way enough time. So you literally have 15 minutes per question. There's going to be 4 questions, and it's not a group assignment. So I don't want you guys doing this with a group. Of course, I have no control over that. But you can use your lecture notes if you would like to, but I would prefer if you guys do this work individually on your own. As I said, I cannot control that. I just have to trust that that's what you'll do. And this will be added to your exam 1 grades. Yeah, so this is the solution because some people did really well in the exam, and they will also have a chance to also improve those grades for them. So this is kind of the solution, and this should get us about what the normal average is for the class. And then you guys will definitely need to study for exam 2, right? And the format is going to be similar. I will reduce the number of questions, not significantly, but it will be a little bit less, and you will have all 3 sections available to you for youth time, so you know what you're dealing with. Any questions about that? And I will post this extra credit announcement after the class, and I will also let you know on Monday. That gives you the weekend to kind of go over your material, of course, and I will let you know when this is available. After this is graded, then Israel and I will have shared office hours where we go over the exam with you. So there's no way for us to return the exam to you, so we will go over the answers to each section. And that's going to be either within office hours or for people who cannot make office hours outside of office hours as well. Okay. Any questions about that?
What's the plan for next midterm?
Pardon?
What's the plan for next midterm? Is it going to be the same format, the same link?
Yes,
and
the format is going to be the
everything?
same for the midterm too. The format is going to be the same as well for the final exam.
And the same link?
The final exam is a little bit longer because you have 3 hours, right? So it's a little bit longer and it's also cumulative, right? For the final
I'm talking
exam.
about second midterm.
The second midterm is going to be, the questions are going to be a little bit less for part B and part C.
Okay, that makes sense.
One question, Professor, is midterm like 2 like cumulative at all?
Midterm 2 is not cumulative. It's just going to cover lecture 7 to 11.
Okay, thank you.
Professor, you said that you would show us the correct answers during office hours.
Yes.
Okay, thank you.
Professor, are we going to be able to dispute some of our questions, like to try to get points back?
So yeah, so you can review the questions. So we can go over that as well in office hours. The thing is, the way that the questions are, the exams are set up with this online format, it's really difficult to really see your exam that you already did unless I print it out and give it to you. So one option is that you will be able to view that exam by coming to the office hours, and I can pull it up and you can look at your answers.
Okay. And cumulative, and then the.
Pardon, I cannot hear. I cannot hear you. Sorry, I cannot hear you.
Sorry. I thought the final was going to be half cumulative and then half the new material that we didn't cover in exam 2.
Yes, that's correct.
Oh, okay. Thank you.
Any other questions?
What is the next midterm covering at 7 through what?
11.
11. Okay, thank you.
Okay.
I'm just wondering, Professor, will there be a curve, or is the curve just the extra credit assignment?
So yeah, that's the extra credit assignment that you're having, yes.
Okay, thank you.
Okay, so as I said, I'll post the slides, I'll post the average slide and also the information about this extra credit assignment after the lecture. And then I'll let you guys know when it's available on Monday to do it. You have, 1 hour. I mean, it'll be available for several hours, so you can decide when you want to take it that day, depending on your schedule. Okay. Okay, so let's continue lecture 8 before, and then hopefully we can get through also most of lecture 9 as well. Yes, Denise, you have a question?
Oh yeah, sorry, just coming from the curve. So depending on how the average is like at the very end of the class, will a curve be considered though, or is it just?
Usually I don't. I'm not sure, and I don't want to answer and say yes or no. I don't usually plan to curve, so I cannot really answer that right now.
Okay. Okay, thank you.
Okay, so in terms of lecture 8, just to recap what we were talking about, right? We talked about eukaryotic transcription and how the transcription initiation is really a complex process involved in many different proteins and subunits that needs to be anchored to the promoter before you can initiate transcription. And this is just a summary of some of those components that we talked about, the order of the components, which I told you guys you'll need to have a good understanding of the different components that gets added and what they do. The last thing we talked about was TF2H, which is the last general transcription factors that gets added to the pre-initiation complex. That general transcription factor have 9 subunits that's split into 2 different complexes. Some of them are involved in protein kinase activity, so that means that they function to phosphorylate the polymerase. And others are involved in some kind of helicase or ATPase activity as well, and helicase really helping to unwind, okay? So the phosphorylation of TF2H is important for the activity of RNA polymerase II. That phosphorylation occurs at the C-terminal domain, and the helicase activity also helps to unwind that transcription start site, forming that open complex at the promoter. So the phosphorylation of the C-terminal domain of RNA polymerase usually exists in two forms. So one of the form, when it's in the unphosphorylated form, is usually referred to as 2A. And 2O is when you have the phosphorylated form. And as I mentioned the last time, this phosphorylation really occurs at the C-terminal domain, and particularly at the serine, the amino acid serine that's present in that C-terminal domain. Okay, so you have a series of serines at position 5, serine 2 at position 2, and serine 7. That's repeated over and over again. And then depending on the stage that you're at in transcription, then different serines will be phosphorylated, right? So the different phosphorylation, the different serines get phosphorylated to either play a role in initiation, elongation, and processing. So this transition from the assembly of the pre-initiation complex to elongation, again, occurs when TF2H is added to the RNA polymerase. Phosphorylation of serine 5 is the modification that creates this phosphorylated state, and that's the serine that gets phosphorylated, okay? This phosphorylation, as I mentioned, is absolutely essential for initiation of transcription and to release that tight bonding of the Tata box binding protein to the Tata box so that the polymerase can start moving along the DNA and transcribing the DNA, okay? During the shift from initiation to elongation, in addition to serine 5, right, that initial serine that gets phosphorylated, serine 2 is also phosphorylated, and sometimes serine 7 can be phosphorylated. It really depends on the promoter, right? So transcription complexes near the promoter that have a C-terminal domain will have this serine 5 that get phosphorylated. This phosphorylation after the initiation, once the polymerase starts elongating, then the serine 5 will then become phosphorylated, okay? And because serine 5 is the serine that gets phosphorylated first and initiate that transcription, that phosphorylation is done by the ATPase activity, or the kinase activity of TF2H. Okay, and this was demonstrated using, again, similar to the protein binding assay that we talked about, right? A gel shift assay or EMSA assay, depending on what you want to refer to it to. That's the assay that showed you that actually TF2H is responsible for shifting from that unphosphorylated polymerase state to the phosphorylated polymerase state. Okay, so you're looking at a gel here. Each of these lanes, or each of the two lanes, have the same general transcription factors, right? They either have additional components, right? F, E, or H, and then you have polymerase that's present in these different sets of samples. You have ATP to help with the phosphorylation if that complex is going to be phosphorylated, to see whether or not the complex is phosphorylated. On the left, you see the different complexes that are formed, right? So phosphorylation, as I mentioned, remember, is 2O, and everything else that's listed below in A state is referred to as the unphosphorylated state of the polymerase. So the first two lanes is with the general transcription factor TF2D, A, and B. In the presence of ATP or in the absence of ATP, you don't see any complex being formed because there's no polymerase in this complex, okay? So there's nothing to phosphorylate to begin with, okay? So the next two, lane 3 and lane 4, you have the same thing, presence of ATP, absence of ATP. You have the general transcription factors, in this case, where you're adding F, and you also have polymerase. And you can see you have that formation of that unphosphorylated state, right? That's indicated here. The same thing in 5 and 6, very similar, except you added general transcription factor E here. And then the last part that you're looking at, last two lanes, 7 and 8, that's where you added general transcription factor H, which is the one that's responsible for phosphorylating serine 5 and creating that phosphorylated state of polymerase. So in the presence of ATP, you have this shift, right, with the phosphorylation. In the absence, you don't have it. So it's not able to perform that, that polymerase conversion to the phosphorylated state, okay? So they did these experiments and they knew that TF2H was responsible for the phosphorylation. And because this was responsible for the phosphorylation, they wanted to see which region in the polymerase is really responsible for that, right? So they hypothesized that, okay, you have this C-terminal domain with these serines. They also knew that serines were the ones that were actually being phosphorylated, okay? So what they did was to determine, is it really the C-terminal domain that's being phosphorylated or some other part of the polymerase that this is occurring? So to do this experiment, again, very similar experiment, protein binding to DNA. Panel A, it's showing you 3 different versions of polymerase. So 2A is the unphosphorylated state. 2B is a version of polymerase that does not, it's a version of polymerase that does not contain the C-terminal domain, okay? So it lacks the C-terminal domain, okay? And then you have polymerase 2O, which is the state that gets phosphorylated for polymerase. So you have these 3 versions, right, of that polymerase. This is just the marker that's showing on the left axis. And then you have all the general transcription factors that are present. And what you see is that you have phosphorylation happening when you have everything that's present. You can see that you have this polymerase. As you increase the amount of the polymerase, you can see that you get a greater intensity of the band, okay? And this is all in the presence of ATP, okay? So you know that so it's in the presence of ATP, so it's being phosphorylated, okay? You don't see any phosphorylation in B because there's no C-terminal domain in this version of the polymerase. It's a mutant polymerase pretty much, okay? You also see in the 2O, which is normally the phosphorylated state. So again, it's phosphorylated, you see an increasing amount. So that's, it's already kind of phosphorylated. So here it's like you would going from a non-phosphorylated state to a phosphorylated state, so it's really better at doing the phosphorylation. Here you've already kind of phosphorylated this version of polymerase and you're just phosphorylating it more. And that's the reason why you see a brighter band here and a weaker band here, okay? But they're both able to be phosphorylated. And the next experiment they wanted to see was like, okay, they knew that you needed to have that C-terminal domain because when you have polymerase missing, you don't have any phosphorylation happening. So they did a very similar experiment, and you can see here that you have, again, all of the subunits plus the polymerase. You have the general transcription factor H in lane 1, right? And you see that you have this phosphorylated state, which is 2O. Lane 2 and 3, What they did here is that they cleaved the C-terminal domain using chymotrypsin, which is a enzyme that cleaves proteins, and it cleaves the C-terminal domain off, so it lacks the C-terminal domain to be phosphorylated. You see the product of the C-terminal domain, right, which gets phosphorylated, but you see that you don't see that conversion of the rest of the polymerase from 2A to 2O, okay, because you cleaved it. And this experiment really demonstrated that it is the C-terminal domain that's being phosphorylated by TF2H, okay? So ATP, so you guys are doing a really good job answering each other's question, right? So you do need the ATP to help to phosphorylate. They provide the phosphates for phosphorylation. So that's for TF2H in terms of the activity to phosphorylate the C-terminal domain, which needs to happen before you can initiate transcription and proceed with elongation. The other activity of TF2H is helicase activity. So it's able to unwind the DNA, which you will need to do, to initiate transcription. You need to form that, that open complex and continue to unwind the DNA. And that's due to some of the subunits in TF2H that have this helicase activity. This experiment is really demonstrating that this TF2H subunit, and in this case it's RAD25, that's referred to one of that particular subunit in TF2H in eukaryotes. And this experiment is really just demonstrating some helicase assay, okay? And the way that the assay works is you have double-stranded DNA. You have one of the strands is labeled with a radioisotope. If you add DNA helicase, which will separate these two strands, you should be able to visualize this on a gel, the strand that's separated from the single strand versus the double strand, okay? So single strand is going to be smaller, and that's what you see here. It's 41 nucleotides because that's what you added. And if it remains together, you see a much larger band, okay? And that's precisely what they did here in this experiment to show that this RAD25, which is a subunit of TF2H, do indeed have this helicase activity. That's also dependent on ATP for energy. So lane 1 is really just the heat denatured substrate, right? So if you denature the substrate by increasing the temperature that we always do, you can see that you have this lower band that you're seeing here, right? So the complex is separated. In lane 2, you have this double-stranded molecule without any protein, right? So that's your negative control. So the band remains high because there's no protein to unwind it, okay? Lane 3 is also the RAD25, so it has the subunit, it has the helicase activity, but there's no ATP, and this activity is dependent on ATP, right? So you don't get this separation as well, okay? In lane 4 and 5, you have RAD25 subunit, but just different amounts, right? So you're increasing the concentration. And you can see as you increase the concentration, you have more of that unwinding occurring, okay? And that's really an experiment to demonstrate that this subunit in the TF2H actually is able to function as a helicase and unwind the DNA. So, and that's for TF2H and the general transcription factors. The way that the TBP, which is a Tata box binding protein, right, which is the subunit that's present in TF2D, and that's what actually binds to that Tata box. Chris, if you look at NMR analysis or any kind of crystallography analysis, you can really see how that protein is connecting with the DNA. So these green kind of look like squiggles. That's the TBP, and then you have your double-stranded DNA, okay? The way that this protein actually connects to the DNA is by really binding, and binds to that Tata box, and it actually bends into groove of the TATA box to be able to bind to it, okay? And it makes a really tight contact with the DNA. So when the polymerase actually gets phosphorylated, this bond is the phosphorylation state of that polymerase forces a little bit of this bond to weaken. And that's how you can kind of shift the polymerase from that position and start moving along the DNA, okay? So it's a really stable interaction in general. It's a universal transcription factor. It's required for all 3 classes of genes, meaning that it's required for transcription of genes that are transcribed by RNA polymerase 1 and also RNA polymerase 3 as well, okay? And it's also present and required in some earlier organisms such as archaea, lots of single cell organisms that don't even have a nuclei. So it's also important for transcription in those particular organisms. In addition to TBP, as we talked about, right, we have the TBP associate factors, the TAFs. There's 13 of them. They're important because they also help to interact with the core promoter elements. They can also interact with other gene-specific transcription factors, right? So these are transcription factors that will either serve as activators or repressors. And when these TAFs are attached to TBP, they help to extend the binding of TF2D beyond the TATA box, right? If you guys remember the DNA's footprinting assay we looked at, you saw that when you have that whole complex, you really go way beyond the +1 site all the way down to +17, right? So you can really extend it beyond the TATA box when you have the whole complex being formed, because it's a huge, and it's also a very large complex. Yeah. Why are the TAFs important in addition to helping to anchor the polymerase? So you guys recall, we also talked about some promoters that don't necessarily have this TATA box, right? So in the case of promoters where there's a TATA box, you have TBP, right? A TATA box binding protein will bind to those promoters. However, for promoters that do not contain that TATA box, these TAFs are important because they're the ones who help the TF2D to bind to other elements within that core promoter. And these elements are either the initiator or the DPE elements within the promoters that these TAFs recognize. They also can recognize promoters that have a GC box, which is other elements within the promoter. And they have 2 activities. So they also have protein kinase activity, so they can help with phosphorylation of RNA polymerase. But they also have this histone acetyltransferase activity, which we'll talk about later on in the lecture when we talk about chromatin structure. But histone acetyltransferase are involved in modifying the chromatin state of the DNA so that you can actually promote transcription. And this is really just a schematic to summarize what we just talked about in terms of the TBP and the role that they play in different types of promoters. So if you have a TATA containing promoter, TBP binds, right? And then it recruits all these other subunits and complexes that's present. And if you have a TATA-less promoter, TBP cannot bind, right? Because there's no TATA box that's present. But instead what you have is the TAFs, which is the subunit number 2, and that's able to recruit TF2D as well. And that's the TAFs is what's recognizing this initiator and DPE element, and you can still initiate transcription that way. In the case of where you have perhaps a GC motif in the promoter, so TATA-less promoter containing GC, again, TBP cannot bind, there's no TATA box, but other components of the that TF2D complex, such as the TAFs, can recognize this GC region and recruit the rest of the complex to the promoter to promote transcription. Okay. And in this case, in addition, so you have, in addition to TAF1 and TAF2 here, you also have TAF4 that's important for this type of promoter. Okay. So this is really just to summarize everything we've talked about in assembling that transcription at the promoter in eukaryotes. I think going through the steps sequentially to determine what's happening at each step is going to be very helpful as we study for this, right? So you know that you need, if your promoter, if the question is you're looking at a data box containing promoter, right, which is more than likely kind of the question that you would get, is you have a data box binding protein that will recognize that, and then recruit the rest of the complex, right? The steps, which one gets added, right? TF2A, you have TF2A, B, you also have E and H, right? And when they get added to that complex, you need to have phosphorylation of this C-terminal domain that's done by TF2H, right? The complex have this open complex, similar to what you saw in bacteria, right? You have these little abortive transcripts that start just to really say, okay, transcription initiation has happened, and you can kind of start transcribing, and these transcripts are just kind of a test run to make sure the polymerase is doing its activity and is able to elongate and move along the DNA to transcribe the rest of the region. And that promoter, and again, once this happens, you have what we refer to as promoter clearance. Everything looks good, and the polymerase can then just continue on to this elongation step, and then you have your transcript being made. You notice that you have, you're going from three serines being phosphorylated, right? Which is serine 5, and then additional serines get phosphorylated as well along the way as you elongate. You have transcription elongation. Okay, so this is kind of really summarizing all of the different steps, right? And I think that this is a good graph to look at to think about the steps what's happening, and also the first summary slide that I talked about at the beginning of the lecture. So some of the things that you want to think about when you're studying for eukaryotic transcription part 1, right? What are the main components? What is POL2? What are the elements within the promoter that some of these components might interact with? What happens if you don't have certain types of elements within the promoter? How does the polymerase get there, right? And also, what is important for the polymerase activity to even begin elongation is really that phosphorylation step that happens at the C-terminal domain. So getting an understanding of what's happening there. And also the role of TBP and TAFs, right? And they're important when you think of promoters where you don't have a data box. So you might either have an initiator element or a downstream element or a GC-rich motif that's present within those promoters. Okay. Again, thinking of which transcription factor is responsible for the phosphorylation. I'm repeating this over and over again. So that should tell you that you're going to see this over and over again in the exam, right? So you really need to know that this transcription factor is responsible for the phosphorylation. The activity is ATP dependent. So in the absence of ATP, right, you will expect you wouldn't have the same activity happening. So just you want to think about it in that context. In terms of some of the fill in the blank types of questions you will see, you already have some experience with these type of questions, but just to briefly, and I remove question number 2, and question number 2 is what you saw in the previous slide, so that's why that's not here. So in the context of in the eukaryotic cells, the assembly of the basal transcription factor usually begins where? And this refers to specificity because, you have to also be very specific with your answers because it's, really, there's just one answer, usually to the questions that I ask. It's very rare that there's multiple answers. Anyone? What is the sequence in the promoter that you have the assembly of these basal transcription factors? We just talked about it extensively, so you guys should remember exactly, right? A Tata sequence, right? You could say a Tata sequence or the Tata box. Either of those would be okay, but the Tata sequence in the promoter is absolutely what's required, right? If you don't have a Tata box in the promoter for TBP to bind, then what happens then if you don't have the Tata box in the promoter? How does the whole machinery get to the promoter if there's no Tata box?
Use TBP and TAF.
You use the TAFs, right? So you then use a TAF, the TAFs will be bound to the TBP and everything gets recruited there. Yeah. Okay, so it's really the TAFs that's binding to those elements and interacting with the TBP. Okay, and what are the two major activities of TF2H that you need to know?
Phosphorylates CDT and unwinds DNA.
And right, and it's helicase activity. Okay. So that's kind of the question that was there before. So before we get into lecture number 9, I also have, and I realized no one told me that I haven't been showing you guys the result of the poll question. I assume you guys can see it in real time with me. So you guys are not even seeing the performance on these polls, which someone mentioned it to me and I didn't realize that. So my apologies, but the reason I look at I give these polls is that the spread of answers is usually all over, right? I mean, and that's something that you also want to keep in mind. So very rare, more than 50% of the participants have the correct answer. So it's a huge spread. I mean, so you really want to think about when you're studying how you will address these questions. So I'm not going to go through all of them because I want to get through some of the lecture, but I want to bring something back that we discussed a few lectures ago, just to see how many of these concepts you're still able to integrate as you move forward in the lecture. So an enhancer is an enhancer blank the gene it affects. So what is the correct answer here? Israel, you're not seeing the poll, right? Are you seeing the changes in answers?
I'm seeing the poll, but I don't see the changes in answers.
Okay, so yeah, that's what I can see it, and I just assume that everyone else can see it. So that's I'm so sorry. So I'm going to try to show that, and hopefully that works. Yeah. Okay. All right, I think it's kind of stable now. Okay, so okay, let's end the poll. And okay, can you guys see the result now?
Yeah, we can.
Okay, so you see the variation in answers. This is kind of consistently how it's been. So this is the kind of question that you will have. So you really want to understand what is happening. Anyone want to give a shot at what the correct answer might be?
A.
B.
Okay, area B. Okay, so
Can be very distant.
It's
Okay.
-60 to -150, so it has to be A, right?
okay, so an enhancer, right? Must it be upstream from the gene it affects?
It can be anywhere as far as I know.
Right. Yeah, it can be anywhere, right? It can be downstream and it can also be within the gene, right? So yeah, it could be upstream, but it doesn't need to be upstream. It could be anywhere, and that's what makes an enhancer kind of unique, right? Because it can be anywhere in the genome. It will still affect that gene that it needs to regulate. Okay. So the correct answer here is B, right? Because it can be very distant from the gene that it needs to affect, and it just loops around and get there. It could be anywhere, pretty much. Okay. It says it cannot be within an intron. That's not correct, right? Because it can be within an intron. It can be within the gene that it's affecting. It doesn't have to be in the same orientation. It can be in any orientation. And there's a whole slide on this, right? Saying that it's orientation independent and also distance independent. And it can be anywhere, location independent. Okay. And it's the same. I mean, A and E is very similar. One is just upstream or downstream. So the correct answer here would be B. Is that clear? Okay. So let me stop that. Oh, I'm so sorry. I wish you guys were able to see all of these previously, but anyway. Okay, so that was just the first half on how eukaryotic transcription, the process of eukaryotic transcription occur, but that's just really the beginning stage, right? So it's a little bit way more complex to regulate gene expression in eukaryotes. And this in addition to RNA polymerase and that pre-initiation complex that gets assembled there at that core promoter, there's other regions within the promoter that's important for regulating gene expression. And that involves transcription activators, okay? And these transcription activators can dictate the starting point and direction of transcription, but they need to, so the general transcription factor will do basal level. These transcription activator comes in and increase that basal level of transcription, okay? And you would need this for many genes, right? If you want a gene needs to be highly expressed, you need to be able to transcribe lots of mRNA for that gene. And that's where these transcription activators, as the name referred to, come into play to help these general transcription factor move beyond that low level of transcription or that basal transcription level. In the cell, these transcription activators are often referred to as gene-specific transcription factors, because depending on the gene you're looking at, different combinations of these transcription factors will function as an activator for that particular gene, or some kind of regulator for that particular gene. Okay, they tend to bind to elements that are referred to as enhancer elements, because again, they're activating transcription, they're enhancing the transcription of that particular gene. They have very specific features. So these transcription activators have domains within the DNA, within the DNA that are important for being able to bind to DNA and interact with other proteins that's present on that DNA, okay? One of the features that's absolutely important that makes them what they are, they need to have a DNA binding domain. So transcription activators or transcription factor needs to bind to DNA. So they need to have a DNA binding domain to recognize the DNA and bind to it. So they always have that DNA binding domain. Usually it makes contact with sequences within that promoter, so the enhancer element or whatever cis element is going to recognize and bind to. And those are usually 4 to 12 nucleotides in length. You also need to have an activation domain always, because they also need to interact with proteins that's already present there, right? So they might interact with one of the TF2Ds or one of the general transcription factors or other types of protein that's important for transcribing that particular gene, okay? Sometimes it has a dimerization domain. So sometimes it have domains where that allows it to form dimers with other proteins or other monomers of the same protein family, okay? Could you just hold 1 second? Let me, you guys can hear that noise in the background? Can you guys hear?
I don't hear anything.
Really?
Are you guys seeing the slides moving actually?
No.
No.
Yeah, sorry for that. Can you see that now?
Yeah.
Okay, could you just hold 1 second, please? Sorry, my apologies. They decided that they should be doing construction right now. Okay, so you could, were you guys able to see this slide where it was talking about the DNA binding domain?
Yes.
Okay, good. Okay, so you need to always have DNA binding domain and the activation domain, which is important. Dimerization, if you want to interact with other protein families or other members within the same transcription activator type family. And you also sometimes can have binding site for certain types of hormones, such as steroids, etc., depending on the activator protein. So this is sometimes. These transcription factors are also classified into different families based on the process that they were found in, and also based on the DNA, really based on the DNA binding motif that's present. Okay, and that really dictates the type of transcription activator or transcription factors. And the DNA binding domain can fall into any of these 3 major groups. So you can have a zinc finger, homeodomain, BZIP, or BHLH. And the transcription activation domain can either be kind of acidic, right? So you have perhaps a lot of glutamic acids or aspartic acids, it's highly acidic, glutamine-rich regions or proline-rich regions present within these transactivation domain. In terms of structurally what they look like, so zinc finger domains, as you can see here, right, you have this alpha helix that's present here, right? Sorry, it's at the parallel beta sheet that's present here, and you also have this alpha helix. You have this zinc ion that's present to help the DNA this domain bind to DNA, but this zinc ion actually does not participate in directly contacting the DNA. Okay, what actually plays a role in the zinc finger being able to interact with the DNA is the actual alpha helices that actually makes contact with the DNA. So you have 3 of these that's usually present, and they can really interact with within the major groove of the DNA, okay, and bind to specific sequences in that DNA. So the amino acids in these alpha helices will recognize the DNA sequence that's present and actually bind to it, okay? So they bind to the major groove of the DNA, and that's really structurally what the zinc finger domains are. Again, they have these 3 combination structure that's present. Homeodomains, the name came from homeobox genes, right? And these are the genes that are usually informed with providing, we always say, extra developmental parts within an organism. So they were first discovered in Drosophila, and these genes tend to transform the body parts. So antennapedia, right? So where the legs are going to grow, where normally you would have an antenna. And that's how they were discovered, because you had these mutants that had these weird physical structure. Okay. And in humans, it's these homeodomains or Hox genes also play a role in some of us or people that we know have these extra digit. Either you can have it on your fingers, on your hands, or on your toes, where you have a sixth digit that's present. And that's really the transcription factor that's responsible for that particular process. They are very kind of similar, right? They also have these helix domains present. And they have 3 of these alpha helix. So 1, 2, and 3, and they kind of twist and turn in such a way that they grab onto, again, to the DNA, binding to the DNA. In this case, different to the zinc finger transcription factor, they actually interact with the minor groove of DNA because it has this extra form, this extra kind of arm that's present that kind of connects with that minor groove of the DNA. Okay, and this is not present in other types of transcription factors. Pretty much, yeah. So homeodomain codes for what they can grow, and also some in, they're present in, even though we're given examples in humans here, they're present in many other types of eukaryotes. So they are homeodomains in plants. They function in many different processes, but a lot of these processes might be involved, again, developmental processes that that the organism might be going through. The last class that I wanted to talk about is the BZIPs, the BZIP transcription factors and the BHLHs. So BZIP transcription factors, and it's usually referred to as BZIPs. And the BZIP, because it has this basic leucine zipper, which is often referred to as this structure, right? And it's called a zipper because you can kind of zip these two monomers apart, or you can zip them together, okay? And these leucine-rich zippers that's present are not actually involved in contacting the DNA. They're just involved in dimers. So these type of transcription factors need to form a dimer, right? And these are the ones that always have a dimerization domain that's present. And what actually contacts the DNA is the basic region of this transcription factor. Do you know why it's the basic region that's contacting the DNA? Why do you think there is? What do you think the reason is?
The DNA is like partially negative.
Right, so DNA is highly negatively charged, right? So that's why this positively charged region is able to attract and actually interact with DNA that's present. And just like, again, with the zinc fingers, where the zinc is there to help the protein get to the DNA, but it actually doesn't bind to the DNA directly. It's the same with these BZIP, where the leucine zippers don't bind to the DNA, but they help to form these dimers that are important for function. BHLH's transcription factors are also proteins that need to form dimers in order to function, right? Very similar to the BZIPs. And they also have their regions that's present that does not necessarily interact with the DNA. So the very basic region is the ones that's actually making contact with the DNA. And both of these are interacting with the major groove of your DNA that's present, okay? So again, just kind of thinking of the fact that these transcription activator have domains that are important. The DNA binding domain and activation domain is absolutely critical for all of them, right? Other types of transcription factors or transcription activator might sometimes have a dimerization domain or a domain for some kind of a hormone signal, but otherwise, keeping in mind that they always need to have an activation domain and a DNA binding domain. This is kind of unique to these types of transcription factors. And of course, like we are able to leverage all of these information that we have learned about how a transcription factor function or a transcription activator work. And I'm using transcription factor and activator interchangeably because they're pretty much the same thing. And we know a lot about how they work. We know that, again, they have this modular structure, right? Meaning that they have parts that are connected that we know about, okay? And if you have a transcription activator, you know that you have two domains that are always present. You have a DNA binding domain, you have an activation domain. The DNA binding domain is required because it needs to bind to a very specific sequence. And that's what's shown here, right? This is an activator sequence, the UAS. This is a promoter that is regulating your beta-galactosidase gene, right? Your favorite gene that you've been learning a lot about, right? And that's driven by the galactose promoter, GAL1 promoter. And you have this, when you have this activation, this transcription activator present, you have high levels of beta-galactosidase, right? So 1800, that's the units, that's how much beta-galactosidase you have. You remove this sequence, this transcription activator cannot bind, you don't have any transcription, right? It's just not gonna happen. And what makes this transcription activator a good system or a good way to determine how a gene, or to understand how a gene is regulated, because the parts are modular, you can remove them from each other, okay? So in this example in C, what you're looking at, the same gene, beta-galactosidase, driven by the galactose 1 promoter. In this region, you have a LEX-A operator. And you guys remember, an operator is bound by a repressor, right, to turn out gene expression, okay? So this is really, imagine that this is a repressor bound to the operator. You shouldn't have any beta-galactosidase being expressed, but you do. And the reason you do is because you're converting this repressor to an activator by just adding the activation domain. So you're really forcing the activation. And we can do that, and that's exactly what we do to understand whether a gene is being regulated by an activator or not, right? We also can look to see whether or not two proteins interact with each other, taking into account the fact that you can separate the activation domain from the DNA binding domain and just stick them wherever you want to. So the way that we use this in molecular biology is in a system in yeast, which is referred to as a yeast-2 hybrid. There's many other versions, but the most basic and common one, and the one that was initially done, is for a yeast-2 hybrid system. It's in yeast, so that's the name yeast, and it's 2 hybrid, meaning that it's taken into account the two component parts of that transcription activator, the DNA binding domain and the activation domain. So usually you have your transcription activator will bind to your sequence in the promoter. It recruits the polymerase and everything there to that promoter, and you can have transcription, right? This is normally what happens. So what we have done is we know that you can separate the DNA binding domain and the activation domain. So what we can do now is add to the DNA binding domain a protein that we want to study, and then to the activation domain another protein we want to study to see whether or not these two proteins are able to interact with each other. How we know that they interact with each other? Because when they come together, they have these two parts, right? So they'll come together, then you will have a full transcription activator that can recruit this basal transcription machinery, and you will have expression of the LAC-Z gene. So we've used this expression of this LAC-Z gene as a reporter to tell us whether or not there's an interaction happening between our protein. And that's what X and Y is. It's two random proteins that we're looking at, okay? So there's many ways, and I've updated these slides as well. I wanted to say that, so I'll post this lecture. I removed a lot of things because it was a lot of information that I think we will probably never get to in an exam, and you don't need to know necessarily to understand the rest of the material in the course. So their way that these eukaryotic activators work, again, they bind to the promoter, they help to recruit and stimulate transcription, they recruit the general transcription factors and RNA polymerase. There's lots of experiments that were done, those are the slides that I removed, to determine whether or not when they assemble on that promoter, are they happening in a stepwise manner that we've seen in previous promoter, where you have the general transcription factors, different units coming in and recruiting everything? Or is it that everything gets recruited already in a complex in this situation? Okay, so in the case of these eukaryotic transcription activator, we know that there's two models, and there's examples of both of these models. So there's no right or wrong way that these are assembled. It just depends on the promoter that you're looking at. Yeah, so certain promoters, you have the assembly where you have the recruitment of the individual components that actually get there by these transcription activators. Okay, so they come in stepwise, or they already assemble, and then just everything gets recruited by TBP down to that promoter. So these are just the two models that we have observed in yeast, and it just depends on the promoter that you're looking at in yeast for these two different versions. What's in addition to these transcription factors recruiting and regulating the promoters are these additional components, which we call mediators. And mediators are usually proteins that can be either a co-activator or a co-repressor, right? So they themselves have no activator or repressor function, but because of what they interact with will determine what their regulatory role might be, okay? So they don't bind to DNA directly, they more serve as a scaffold to help to recruit a number of other proteins and have everything present within the promoter. They can interact and modify chromatin, because you need to have the chromatin modified in order for you to be able to express the gene, right? The chromatin needs to be a little bit open or accessible, and they help to recruit some of the enzymes that's involved in changing that state of the chromatin. Okay, and this is really a summary of that, right? Where you have these enhancer elements, where you have some activators, you have other transcription factors that are at distal sites, you have co-activators that can be present, you have your basal transcription factor, your general transcription factor with the RNA polymerase that's present at the co-promoter. And all of these proteins kind of need to interact with each other and really be present at that promoter to regulate transcription. And they get there by the help of these mediators, right? Which really help to glue all of these different parts together within the cell. This mediator complex, which we're not going to get into, is a really large protein complex. So it has like 25 to 30 different subunits, depending on whether you're looking at yeast or humans. But it's a huge complex that's really involved in bridging all the different protein interactions at the promoters of the gene that they're regulating. Okay, so that's just to summarize again. I'm going to summarize what we talked about: eukaryotic transcription, RNA polymerase, general transcription factors. We have these sequence-specific DNA binding transcription factors, which need to have two important structures. We talked about co-activators and co-repressors, which are part of that mediator complex, and knowing the different cis elements that these both the general transcription factors bind to, and also these activators. Again, they're site-specific for the transcription activators, and understanding the whole promoter region, right? What's happening at the core promoter, what's happening at the proximal promoter, right? And then you also have these distal promoter elements where you can have enhancer regions as well present. So that's kind of just basic transcription that's happening at the promoter of our gene. But for any of those things to happen, we have to deal with the actual structure of the DNA. okay, and how it's present within the cell. So eukaryotic genes, this is not true in bacteria. Eukaryotic genes are not naked, right? So the DNA is just not lying around there, present within the cell, and polymerase can get to it, or transcription activators can get to it. It's really compacted because it's a lot of DNA. Okay, so if you were to stretch the human DNA out, the length would be about 6 feet. Yeah, if you stick every part of every DNA, if you were to lay every DNA molecule end to end from the human genome, you would literally reach to the sun and back hundreds of times, right? It's a really, really large amount of DNA that we have. And that amount of DNA needs to be compacted into a very tiny nucleus, okay? And the way that that happens is with the help of a whole range of proteins that really help to wind the DNA in such a way where it's completely compacted, okay? And that's the only way, I mean, it needs to get to that state before it can go, before it is able to go through mitosis, right? Because again, nucleus is really, really small. So what helps to compact the chromatin, what helps compact the DNA, or the way the DNA is compacted, is usually referred to as chromatin, right? And that's the DNA and all the different proteins that help for this process to work. The main players are histone proteins, but you also have additional non-histone proteins that also help with this compaction, okay? Depending on the level of chromatin compaction we're looking at, you can have many different stages. So this is the stage it needs to get to before you can go through mitosis, right? So it needs to be compacted like really, really significantly. So you're going from a normal naked DNA, like we've been talking about when we talk about, when we draw DNA double helix, etc. This is kind of what we're talking about, but we know now that the DNA is really compacted with the help of these histone proteins and different higher order structure to get it to this really compacted state to go through mitosis. If you were to look under an electron microscope, right, you can see that these structures are actually what's happening. So you have, in this case, a 30 nanometer chromatin structure, which looks like this one, right? So you can see kind of, in at least you can see that they look kind of compacted compared to B, right, where it's less compacted, right? So you see the individual nucleosomes, right, next to each other, okay? So you can look at a different stage. Heterochromatic regions are regions where the DNA is not accessible. Euchromatic regions are often regions where you have a lot of genes, so you need to have this structure to be a little bit less compacted so that the polymerase can access it and all these transcription factors can access it, right? So it's a little bit on a more loose. And that's really the difference between these two states. We talked about transposons and how transposons are in our cells, right? And we don't want transposons to be active and turned on. In the heterochromatic regions, which is highly compacted, there's a lot of transposons because you want to kind of hide them, right? You want to protect the cells, so you kind of hide them in these heterochromatic regions. And that's often where you find a lot of transposons being present. So what makes up a core nucleosome, right? So one of these little beads that we're looking at is you have about 146 DNA base pair of DNA wrapped around a set of proteins, and these are called histone proteins. It's wrapped around 1.6 times around these histone proteins, and it also includes linker regions, right? So you have 20 to 60 base pairs of a linker region that's present, okay? So 146, 20 to 60, this is what makes up a nucleosome unit. What are these proteins, these histone proteins? So these histone proteins, there's 4 that's present. They're highly conserved proteins across eukaryotes. And depending on how they are interacting, they combine together to form what we call a histone octamer, okay? So this includes H2A and H2B, which forms 2 heterodimers, right? So you have this dimer and 2 times, right? So you have each of those here. And then you have an H3 and H4, which form a tetramer. Okay, so you have the 4 components here. So 4 units, but the way they come together forms this histone ultimer. That is what makes up that histone core of proteins. These, again, highly conserved, very little variation across eukaryotes. If you look between even P and perhaps humans, there might be 1 base pair change between them. That's how conserved these proteins are. And what is special about them, how they're able to regulate the compaction of the DNA, is due to these tails that are sticking out. So they have their proteins, they have orientation. So you have an N-terminus and a C-terminus. So the amino terminus is usually referred to as the N-terminus, and then you have a carboxyl terminus, right? These N-terminal tails that stick out from this nucleosome, these tails get modified, okay? And this modification of these tails is really what helps to regulate the compaction of that DNA. Okay? The way that they interact with the DNA, so they are able to interact with the minor groove of the DNA at regions which are often AT or AT rich, yeah? This is preferred, and this probably should make sense because you have less bonds that you're forming. So it's easy to kind of manipulate that, you know, to unwind or shift the interaction between these proteins and that DNA, okay? In addition to the core histone, you have these histone linker proteins. So you have these histone, these H1 proteins. They're also highly conserved, but they can be a little bit more variable across eukaryotes. But you have these histone proteins that also help. They're important for tightening the DNA around the histone proteins, but they also help to define the way the DNA is wrapped around the histone protein and also gets removed, right? So they help to provide a more defined angle of DNA entry and exit on the nucleosome. So they're very important within the cell. And this is if you look at how those nucleosomes are compacted and how they're able to regulate gene expression, right? Or the effects they have on gene activity, for example. So in this case, you have this highly compacted 30 nanometer fiber. In this state, it's you have a repressed state, right? Because you have your nucleosome, you have your DNA, you have your H1. The transcription start site is not accessible, okay? It's not accessible because it's wrapped up with all of these proteins and also the linker protein that's present. This becomes competent when you start loosening, removing these histone proteins from being present, right? So you have your histone, the linker histone proteins. So you have your core proteins, DNA is accessible, an activator can come in, bind to the DNA, recruit everything that needs to be recruited, and you can have transcription. The other way that this also happens is if you have, for instance, maybe it's not the histone H1 that's blocking the transcription, but an actual core protein. So the core protein is on top of this transcription start group site, right, which is shown by this arrow. It's not accessible, right? Even if you remove the H1 protein, it's still not accessible, so it's still in a repressed state. However, you can evict this histone protein, right? So you can just kick it out and add an activator, or the activator kicks it out, and then it's able to perform transcription, and the gene becomes active. So the way that this is done is through a set of proteins called chromatin remodeling complexes. These proteins are highly ATP dependent, and they help to change the interaction between the histones and the DNA. By doing so, they're able to help the transcription factors, such as transcription activators, right, bind to the DNA elements and recruit all of the transcription machinery. The way they do it depends on the complex. So some chromatin remodeling complex will be involved in nucleosome sliding. So it will slide the nucleosome, remodel the nucleosome, displace the nucleosome, or completely replace the nucleosome, okay? And one of the very, very common nucleosome remodeling complex, again, this is a complex, so there's many, many proteins that are part of these complexes, okay? I mean, every couple of months, there's a new paper on a new member of these nucleosome remodeling complexes that have been identified, right? That's to say, you know, how complex it is, we still don't know each of the players that's involved, okay? And this is why SNF is usually referred to as switch SNF. They were initially discovered in yeast because they're involved in mating type, the mating type in yeast and switching of mating type in yeast, and also in sugar fermentation, right? So SNF just stands for sucrose non-fermenting. So the mutants have a problem, a defect in fermenting sucrose. So these huge family of nucleosome remodeling complexes involved in nucleosome displacement. It's ATP dependent. They need ATP in order for this to work. So you can see if you have your DNA wrapped around your histone octamer, this complex comes in most often by modifying those histone tails we talked about. And by doing so, it's able to either change the way the DNA is wrapped around those histones, those core histones. So you can see this is the normal state. It comes in and it kind of slide it through the two histones, right? By doing so, DNA which you cannot see, which is on the other side, is now accessible. Okay, so it's making it accessible. Or it can completely unwrap the DNA from the histones for a certain period of time so the transcription of that gene can occur. Okay. The other group of histone remodeling complex is this is also, again, ISHRI, just depends on, you know, how you want to pronounce it, or ISWI. These are chromatin remodeling complex that slides the histone octamer along the DNA. Okay, so you have your histone, also ATP dependent, and it really just slides it out and make a region that was hidden by these histone now make it accessible. So you can actually access the DNA that's here, recruit a transcription activator, and transcription can occur. Okay. This is the last one I'll talk about before and then I'll stop here. But because these are three major ones that you need to know. The other major group of chromatin remodeling complex is SWR1 chromatin remodeling complex. And these are involved in replacing the core histone with a different version of that. And a different version is usually referred to as a histone variant or a variant histone. Structurally, it's very similar. So H2A and H2B, the variant in this case would be an H2AZ, which is the variant of H2A, that will interact with H2B. By replacing this core histone with this variant, the way the DNA is wrapped around is much looser. The interaction is not as strong. So because of that, this region becomes a little bit less, or becomes destabilized. Okay, so the DNA is much more accessible to the transcription machinery. Okay, so that's the other way that you can modify the chromatin. But I'll stop there before we go into methods, and I'll try to reinforce some of this. I'll be here until 2, so I can answer questions.
Thank you, Professor.
Yeah, it's a lot of material, a lot of information that I want to get through. So I'm sorry if I rush through some of this, but feel free to ask questions.
I'm just curious, what's the grading scale for this class, or is that determined by the end of the
That's determined.
course?
There's no, yeah, I don't have that deeper. That's going to depend on the end of the end of the class.
Okay, so like, because a lot of us are still concerned, like, because we're worried if there's like gonna be like a regular grading scale, like 90 to 100 is an A, like 80 to 90 is a B, stuff like that.
So yes, I don't have an answer for what the grading scale is. So yeah, it really depends on the end of the class, yeah.
Okay, and just one more thing, could we like further sort of discuss the midterm to like exam format? Because a lot of us are still concerned about like, even though it's gonna be like less questions, we still have some like questions and concerns about the format.
What exactly, I mean, the format is the format, and that's the way it's always been. The other molecular biology sections that also have the same format. So that's the format of this type of class and type of material. So the difference really is just how many more questions you have in each of the different sections, but that's really is the format of the exam.
Okay, thank you, Professor.
Take care.
Thank you.
See you Friday.
Bye. See you.